Actually, I studied geoproteins before I retired last year, and then I am very glad to be here to learn more about the GO and to connect the families. Then I didn't notice that before in Korea. Actually, I was a student Lutz Bombaum who cloned the geoproteins a long time ago, but I didn't have a chance to study on this one because there are excellent German postdocs in his laboratory. And so when I went back to Korea, then I had a chance to look at this protein, and then I realized that this must be the new way to improve the neurological diseases. So the reason is, the reason I started to work on G protein is that G is very abundant. We all know that. And it is very difficult to study in animal models. And there is no direct way to measure the geospecific functions in vitro and in vivo. Although we heard a lot of the interactions this morning, but GO does not have any enzymatic activity unlike GI proteins. So we address these challenges using two approaches. First, we use the different conditional knockout method in the cerebellum. The deficit in the cerebellum does not cause any epileptic seizures. And also, if we cannot measure the GO's activity, why not looking at the interactive partners? So we tried these two approaches. So this one just slowly shows that how much G protein is enriched in the brain. So in the middle, the GO is one of the top 40 proteins enriched in the brain compared to the liver proteomics. And also this is enriched in the synapse. And when we look at the GIO-coupled receptor in the cerebellum, which is the cannabinoid receptors. Cannabinoid is the active components of the medicinal plant like marijuana. And when the Prethner and colleagues treated the brains with this agonist Wnt compound of the CB1R, they found that the GO is dramatically activated, and compared to that one, GI is very minor. So I think cerebellum is the best place to look at the GO's function with respect to its GPCR, cannabinoid receptor. So I'll come back to this one. The original slides, I included this earlier, so I come back to that slide later. So what we did is we isolate the synaptosomal fractions from the red cerebellum and then look at the immunoprecipitation profile. So we used the CB1R antibody and to precipitate it and look at the GO interaction there. And then, this works, okay. So the red arrow points that the GO interact with the CB1R proteins, and the Wnt complex, which is agonist, does not change the interaction. However, other antagonists, like AM or SR compounds, can disrupt this interaction. And GTP gamma S, of course, disrupts this interaction. And shows that the GO may interact with the CB1R. But as we know that the immunoprecipitation causes a lot of artifacts. So what we did is we purified the G proteins using G proteins as a fusion protein to GST, and we make the active form of G protein incubating in the GTP gamma S, and inactive form of the G protein with the GT DP. When we look at the, okay, so this one, the pointer does not work. And, this should be closer, okay. Thank you. Oh, this is the pointer?
Yeah. You can't see it, it's electrical. Just hold it on.
Okay. So when the G beta interaction just validate the GDP form, inactive form of the G proteins. And when I pointed that the CB1R interacts with the purified inactive form of the GO proteins in here. So we have a basis to go forward to the cerebellum specific GO knockout. Yeah. So this is quite complicated. So because I have an original slide, so I thought the students may have the original slide, so that's why I still keep the original one here. So this is how we obtain the cerebellum specific GO knockout mice. So cerebellum has a great cytoarchitecture. We can identify the synaptic regions. And here, so the box shows the synaptic area of the presynaptic granular cell neurons, which is green, and the postsynaptic, presynaptic granular cell neurons, and the red one is the postsynaptic, the Purkinje cells. So when we look at this one, the CB1 is located in the presynaptic nerve terminal. So we want to knock out the GO proteins in the same neuron terminal where CB1R is located. So we use the glee mouse. Yes, glee mouse. Glee is actively expressed in the proliferating progenitor cells of these green cells. And so, but even if we use the glee mouse, the deletion is not sufficient. Yeah, deletion is not sufficient. So what we did is we used the double knockout system with the constitutively deleted GO allele and the conditional allele in here. And in that case, we can lower the GO proteins in these mice, because once GO protein is translated, it's really difficult to delete it or lower the protein level in the post-mitotic neurons. We have the same experience with CAMK2. GO is disappear 6 weeks after the CRE is activated. So in this system, we can efficiently delete the GO proteins in the postnatal green neuron. Green means granular cell neurons. So in here, we can lower the G proteins, and this disappearance is only found in the distal region. Sorry about that. Not familiar. Distal region of the molecular layer, proximal region, molecular layer, which means that the neurons in here is formed before tamoxifen is injected. The nerve terminal in the superficial layer is formed after the tamoxifen injected. So we can specifically delete the presynaptic nerve terminal after the tamoxifen administration. Okay. So this is the quantification of the GO deletion. In here we can delete the GO proteins, but somehow G beta is also reduced. So when in this mouse model, when we don't have the GO, there is also a G beta decreased. and we think that there is no excessive G beta gamma formed in this mouse model. So whatever we found is maybe is the ascribed to the geospecific deletion rather than excess amount of G beta gamma. And also in here, we have the geoimmunoreactivity in the superficial layer is lowered in the mutant mouse. Okay, now I know how to do it. Okay. Okay, so we looked at the synapse formation in the superficial and the deep layer in here. So we measure the number of the synapse between the the presynaptic and the postsynaptic neurons in here. And when in the mutant mouse, this synapse formation is reduced, which is shown in the left. So we measure the spontaneous excitability of the postsynaptic Purkinje cells in here. And then both the frequency and the amplitude is increased in the mutant mouse, as you can see in the red data. And we also look at the suppression, any changes in the DSE. DSE is a very manifest phenotype of the CBR1 in the cerebellum. So DSE is the depolarization. So once postsynaptic depolarization induces the secretion of the endocannabinoid from the Purkinje cells, and which can go back to the presynaptic neurons, and then it binds to the cannabinoid receptor CB1R, and then activates the GIO, and this which suppresses the neurotransmission from the presynaptic nerve terminal. That is found as this is the depression of the induced suppression of excitation, this DSE. But in the mutant mouse, this DSE is reduced, suggesting that the CBR1 and the GO is very important in this short-term plasticity, and the GO plays a key role. Okay, then we looked at the behavioral phenotype of these mice, and then as we hypothesized that the cerebellospecific knockout does not induce the lethality, so the size and the body weight and the locomotion are similar between wild type and the mutant. And unsurprisingly, we found that there is no change in the eye movement. There's no lack of eye movement. So eye movement is a manifest, nystagmus is a manifest phenotype of the cerebellar dysfunction, but which is absent in these mice. So when we look at the brain area, and then in here, in here, on the right side, the F1, F2, you can see the GO is not deleted. The lack of the red signal means that GO remains there. So in this lobule, the residual GO plays a role, and there is no defects in eye movement. So it's further strengthen our hypothesis. So this lobule is generated, synapse in this lobule generated before tamoxifen administration. Very ancient lobule of the cerebellum. That's why GO remains there. We don't have any defects in eye movements. We measure the hanging wire grip strength using hanging wire strength, and also footprint analysis here. And there is a change in the mutant, which is shown in the red bar, and also which consistently in the footfall errors are increased in the mutant mouse. On the G, graph G, means that this mice show the ataxic-like behavior, and there is compromised posture control. We also measure the learning ability. So we did the rotarod rest test, and wild-type mice gained the full level of the retention, means that they learn over 5-day period. But however, mutant mice does not learn to retain on the rolling rotarod, suggesting that this GO plays important role in the cerebellar dependent motor learning and the behavior. So then we ask the questions, if CB1 and the GO is important, how their signals lead to the reduced release of the neurotransmitters, such as the DSE. So we looked at the snare complexes, whether they can interact with the G proteins. In here on the right side, this is the immunoprecipitation test. Then immunoprecipitated with the GO antibody, and then we found that there are a lot of proteins, snare complex proteins interact with GO. Well, I believe, I don't remember, but I believe that only 2 of them are indirect interaction, and the other one is direct interaction when we verified with the purified GST pull-down assays. So with this proteomic data, we did the IPA analysis, and then when we look at the GO is the central part. GO is the central part, interacting so many different proteins and in the synaptic area. So I stopped in here because there is no way for me to move forward. But since AI is prevented, we can define the, estimate the protein structures, and so it's not efficient to pursue the GST pull-down assay or IP co-immunoprecipitation or something like that. So we better estimate the potential interaction. Why geoprotein interacts so many different proteins? It interacts with the pKa as well. But that interaction does not alter the enzymatic activity of the protein kinase A. So why geoprotein interacts so many proteins, and then I surely do not know how to solve that problem. Some of you may have an idea to look at the interaction with respect to the human mutations, not just only look at the geoproteins, but whether the mutation may change the interacting partners or something like that. So I brought this recent data about the epileptic seizures about the GO knockout mouse in the forebrain. So we used different mice, EMX1 Cree, then that also kills the, leads to the lethality. So about before the mice become mature, they almost died. So what we did is we found that in the previous slides, the GO interacts with the SV2A. SV2A is a target molecule of the anti-convulsant drug, levetiracetam. We heard that before. And then so-called Keppra. This drug is used by a lot of neurosurgeons after they dissect out the brains, and then that is very powerful epileptic, anti-epileptic drugs. So since GO is interacting with the SV2A, we did the slice culture and measured the spontaneous epileptic activity in here, and which can be blocked by the levetiracetam in here. Also, treatment of this drug of the animals can extend the survival in here. So this can be a very good reference drug. When we screen the potential target drugs, then there should be some reference drugs which can block the epileptic or excitability. So this can provide as good reference drugs. Okay, so far I'd like to start my talking to make a conclusion. And so this one said, through this study, we know that the GO regulate the retrograde signaling of the endocannabinoid and the cannabinoid receptor pathway by inhibiting the synaptic vesicle excitosis and controlling the DSC and Purkinje cells. So, but our study shows that the GO may be important to control the brain homeostasis and the balances through the cannabinoid receptor. This cannabinoid receptor is very well known in the hippocampal neurons as well. But our study has a limitation because we knock out the GO proteins in the protein. But I heard that a lot of human patients, they have the heterozygote mutations, which is more like the gain of functions compared to our study, which is the loss of function studies. Then I'd like to stop here and then happy to take questions and comments.